So we're going to talk in this set of notes about some of the tools we use in biology, mostly focusing on microscopes. Um, so microscopes were really um, invented, I would guess you would say, in like the 1600s. What was it? What was necessary before microscopes could actually be invented? There's one key piece of technology that microscopes require um, that started to be developed around this time. You know what that would be? Ash? No, because actually the first microscopes used natural light. They used sunlight. They often use like a mirror to like focus the light on the specimen. It's a good idea though. It was something else. Michael? Yeah, lenses. They needed lenses. That was what was required because that's really what a microscope is. It's either a single or a combination of lenses. A magnifying glass is a lens. Telescopes use lenses. And so lenses weren't really developed until this time around the 1600s. And so that's what allowed for scientists to start to develop microscopes. The first microscopes looked kind of weird compared to what you would imagine. This was one of the first microscopes, this picture here. Like, what is this thing? How would you actually use this? Why is it made of metal? So the way this microscope works is this was the type of microscope that Anton van Leeuwenhoek invented. He was like really important in the history. He made these super powerful microscopes back in the mid 1600s. And actually, he was very secretive. He wouldn't tell anybody how he was making these lenses. And people tried to figure it out because they were really powerful. They could magnify images up to like 200 or 300 times using this technology in the 1600s. So the way this worked is the lens is actually here. It's like this tiny little lens, a little piece of glass that's here. And on this like needle thing, you would put whatever specimen and you would look at this microscope from the opposite side that we can't see. And so you kind of hold it up to the light. You'd be looking through this tiny hole, which had like this little glass lens embedded in it. You could move the specimen by turning these little knobs. You can move it forward or back to focus. You could turn this knob to raise or lower the specimen. And so there were, he produced many of these microscopes, but was very secretive. Like I said, there's 9 of them are still around. Eventually, people learned that the way he was making these lenses, he was melting glass and allowing it to drop into water where it would form a very small sphere, which was making a lens of very strong, a very powerful lens that could magnify a lot. But people didn't figure that out, and he wouldn't tell anybody either. So once these microscopes started to be developed, people started looking at all sorts of different things. And here is one of the famous. This is by Robert Hooke, another early scientist that looked at things through the microscope. And he wrote this book, and he drew these detailed drawings of things that he saw through the microscope. Things like this. This is one of the drawings that he made, and this was like a bestseller. This book of illustrations, because people didn't. The everyday person never would have looked through a microscope. Would have no idea what these very small organisms and things would look like. Robert Hooke. This is the kind of microscope he was using. This looks probably more familiar to you. You can imagine. Okay, there's a lens up here. There's a lens down here to magnify the image. So today, our modern microscopes are, we have several. And the microscope you'll be using is the compound microscope usually. And these are, you guys use these 7th grade, right? All right. So this is review, but these are compound microscopes. They have a lens here in the eyepiece, and you have a lens here in the nose piece that you can select different powers of. And so they both are magnifying the image at the same time. So it's like this lens magnifies the image a certain amount, and then the light gets to this lens and it magnifies it even more. And so that's why these compound microscopes can give a pretty high magnification. Why is their color so bad? So because that's the way these compound microscopes work, the total amount that the image is being magnified is the power of the objective lens multiplied by the power of the eyepiece lens, because they're double magnifying. Now, if you go to college and use a microscope, they might have microscopes which magnify an image like 1,000 times or 1,500 times. These microscopes that we use magnify a maximum of 400 times. So depends, there's different types of microscopes, different methods, but they go up to about 1,500 times. When you look through the microscope, the image you see is actually backwards and flipped upside down. Because of the way the lenses in the microscope works, the image is inverted and flipped backwards. So here's what it looks like. You see a very flat image. When you look through the compound microscope, it's a two-dimensional image. Also, the things you look at have to be very thin. Because of the way the microscope works, light needs to pass through your sample to get to your eye. So you can only look at very thin things. You can't look at your hand under these compound microscopes because light can't pass through your hand. The dissecting microscope works a little differently. The dissecting microscope sometimes called a binocular microscope, works in a little bit different way. So this dissecting microscope, it uses light coming from above, which bounces off of the object and then comes through the lenses. So you can look at opaque objects which light cannot pass through. Also, because it has two eyepieces, it gives you a three-dimensional view of the image. You need two eyepieces to get a three-dimensional view. So you can see depth of an object much better using this type of microscope. Usually this is less powerful. Some of our dissecting microscopes, this one magnifies 10x or 30 times. That's the maximum. So much less than the 400 times magnification you could get with a compound microscope. So this is like, these are images you can see a dead bee through the dissecting microscope. And like you said, it's a three-dimensional image. You could see the detail. You could see sort of the depth as you're looking through that. And there's a spider. So we're going to use microscopes today in our second block. And so we're going to review the procedures or how to do that, because whenever we do, whenever we do a microscope lab, oftentimes people can't find an object or they're having trouble focusing. So it's really, it takes a little practice, but once you know the correct procedure, it's not too difficult. So we'll review that and then you can practice it today. So these are pretty expensive, so we want to be careful with them. I don't want to drop them. Carry them with 2 hands if you would. You can clean. Sometimes, people touch the eyepiece and they get their grubby fingers on the objective lens and it gets grimy. You can clean it with special paper that we have. It's called lens paper, just to clear off any grime that might be on those lenses, because that makes it difficult to see things when you have to look through that. When you're looking at an object, you always start on the lowest power. So these objective lenses rotate, and there's different powers. The shortest one is the low power objective. It says on it 4 because it magnifies 4 times. The other objectives we have on our microscopes are the 10x objective, and then the high power 40x objective, the longest one. So you could rotate through. You have to make sure they're like clicked in. You could probably hear that. Sometimes people like switch to one and it's not quite clicked in and they don't see anything. So you just want to make sure it's clicked in and in place. So to see something, you always start at lowest power. There's a dial here which changes the amount of light. It's got these diaphragms work because there's a little plastic piece under there with holes of different sizes. And so one of those holes is really small and doesn't allow a lot of light through. One of them is larger and allows more light to come through. So you always want to start with the lowest setting, and you can increase it because as you zoom in, you need more light. You put your slide, so you always are looking at something that's on a slide here, and you put your slide on the stage, which is this flat black piece. And what you'll see is there's 2 different types of stages that we have. 2 different ways of attaching your slide. Like this is the simple one. You just like put the slide under these clips, and then you got to move the slide around. You got to move it with your hand. Right? This is simple. It doesn't break either. This microscope works differently. What you see here on this microscope, it has this like contraption here. That's called a mechanical stage. It's got a little clip that's spring-loaded that keeps the slide in place. And then to actually move the slide, you have these knobs that you turn. Rather than moving it with your fingers, one knob moves left and right, the other moves forward and back, and it just allows you to really have good control over the slide. When you're using your fingers, you probably remember like you move it a tiny amount and it flies off the field of view. With these knobs on the mechanical stage, it's much easier to just move things very carefully. So I like this kind. Problem is they break easy and things like that. So most like professional people using microscopes would use something like this with a mechanical stage. You focus the microscope using these knobs here. The larger one is called the coarse adjustment. It moves the stage a lot to focus a lot. The fine adjustment's the smaller knob. It just moves things really, really small amounts. It's for sort of fine tuning the focus. So the coarse adjustment moves it a lot. So you put your slide on there. Make sure it's centered. Look through the microscope. Turn the coarse adjustment until you can see the image, until you can see the sample you have. You can fine tune it using fine adjustment, and you want to make sure it's centered. Okay, so either using the mechanical stage or using your fingers, you want your specimen right in the middle of your field of view. If you want, then you could switch. You keep everything the way it is. You have it focused and centered under low power, then you could switch to medium power. Don't touch anything else. When you switch, it should still be in focus or really, really close. So then you could focus again if it needs some small adjustments, and you'll see it on medium power. Again, you want to get it centered before you switch power, and then you could switch to high power if you need to. As you go to high power, the image might become darker, so you could open up the diaphragm so more light's coming through. Once you are on high power, typically when it's in focus, the stage is going to be really close to the objective lens. And if you use the coarse adjustment, you could smash the objective into the slide. So once you're on high power, you're only going to use the small knob, the fine adjustment, to kind of make sure it's in focus and that things are clear. Now, it's sometimes harder to find the object under high power. Once you switch, sometimes people lose it. If you do, you just go back. You go back to medium, find the specimen, focus, center, then go to high to try to find it again. You always are going to go, if you lose something, go back and start back at medium or low power. So we look at things on slides, as I said. Now, sometimes the slides are already prepared for you. Somebody put a sample of whatever bacteria on there and glued it together. They're prepared slides, so it's all prepared for you. Other times you need to make your own slide. You do that using what's called a wet mount. Basically, you have a blank glass slide, put a couple drops of water on it, put your sample in it, and you cover it with a little plastic or glass cover slip. And that sort of seals it all together. It's held together by the cohesion of the water, and then you can look at your sample. Some of the things we look at in the microscope don't have any color to them. They're kind of transparent. In that case, we often might stain a specimen, which is using a special chemical that the cell or the specimen will take up, and it allows things to stand out more, and you could see them more clearly. Some stains, for example, will stain the nucleus of a cell darker, so you could look at the nucleus. Others might stain the cell wall different colors. So we might use stains to help us see things more clearly. So again, this is what the traditional stage cups look like, and this is the mechanical stage. Sometimes the controls are underneath like this. Our microscopes have two adjustment knobs separate from each other, the coarse and fine adjustment. There's some microscopes where it has two knobs in one, a larger knob on the outside and then a smaller fine adjustment on the inside, like that. To determine magnification, remember the eyepiece power times the objective lens power. So for example, 5x eyepiece, 40x objective gives 200x total magnification. That means the image will appear 200 times larger than its actual size. It's been magnified 200 times. When we're measuring things, we often will use different units. So millimeters, which you're probably familiar with, when you look on a ruler, the smallest line usually is millimeters. But in measuring things in the microscope, often they're really, really small. And so then we might use micrometers. This is the symbol for micrometers. It's the Greek symbol mu. Looks kind of like a cursive U. And there's actually 1,000 micrometers in a single millimeter. So micrometer is really small. So in this ruler, each of these small lines represents a millimeter. And within that millimeter, if you split it up 1,000 times, that's a micrometer. So if you have millimeters and you want to know how many micrometers, just multiply by 1,000. If you have micrometers and you want to convert it to millimeters, you divide by 1,000. You will measure some things with your microscope today. So to do that, what you're going to do is you're going to measure the field of view. The circle you see when you look in the microscope is called the field of view. And low power has the largest field of view. So that makes sense. At low power, you could see a wide area. As you switch to medium power, you can see less. As you switch to high power, you see less because you're focusing on a small area. Just like if you imagine just looking with your eyes on the parking lot, you can see the whole parking lot. If you were to look with like some binoculars, you can maybe zoom in on that house over there. If you switch to like a telescope with more magnification, you could zoom in on a specific window. but you're seeing less and less of an area. It's just enlarged each time. So we can put a ruler under the microscope, and you'll do this afternoon, and actually measure. And see, when you look at low power, how wide is the field of view actually? Okay? How many millimeters wide? How many micrometers wide? And then you could repeat that same procedure under medium and high power. So sometimes you will have to estimate the size of an object. So let's say we look in our microscope, we put a ruler under there, and we find that this is what we see. So the field of view in this power, it's about 2 millimeters, 1 millimeter, and then 2 millimeters here. So if I'm looking at this pollen grain here, how big would you say it is? How big is that pollen grain? This is 1 millimeter, and this is under the same power. So that's 1 millimeter, maybe half. Okay. So you can measure that by estimating. What you could also do is you can take the field of view. So the field of view is 2 millimeters, and you can kind of estimate how many of these pollen grains would fit across here. And if you do that, you say, well, maybe we could fit 5. The field of view is 2, divided by how many fit across would give you 0.4 millimeters. And again, these are estimates. Now, let's just finish up here. Electron microscopes. Do you guys have this slide? Yeah. So there's another type of microscope called an electron microscope. We don't have one here. They cost probably tens of thousands of dollars. But they're pretty cool. They have one at Whitesboro High School. We don't have one though. And an electron microscope, instead of using visible light, uses a beam of electrons to make an image. These were developed in the 1930s, and you could get an amazing level of magnification using electron microscope. You can get up to, millions of times magnification. Has very high resolution. Here's what a modern electron microscope looks like. There's a couple types, and they work in sort of different ways. A transmission electron microscope works like this microscope, except it's using electrons rather than visible light. Gives really high magnification. Okay, you can see very, very small things. But again, the specimen needs to be thin enough to allow electrons to pass through. The scanning electron microscope gives really cool images like this. It basically scans across the specimen with this beam of electrons and lets the electrons bounce off to form a three-dimensional view of the image. Problem is, you can't look at live specimens using this. Typically, they need to be coated in metal, and they're typically dead in order to do that. Okay, the last two tools we'll talk about are tools that we'll use later on in the year that help us take a mixture of molecules and separate them by their size. And you may have done this in 8th grade. I don't know if you did chromatography. So chromatography, here's my pictures. What you do, oh, here it is you place a sample of some liquid with a mixture of pigments in it. And then you hang the paper so that the bottom edge is in a solvent. And the solvent will work its way up the paper. But as it does so, the pigments will separate out. And they'll form a bands of different colors. Did you guys do that in 8th grade? Yeah. No, some people did. Ash, you did it? Yeah. So like these, sometimes you do it with markers. Like a black marker actually has several colors in it, and so as the solvent soaks those pigments up the paper, the different colors that are used to make black are separated out. Another way of doing that with DNA. Oh, here's another example. This is showing plant pigments. So if you smash up some leaves of a plant and do chromatography, the actual pigments that are in those leaves separate out by different bands. You could also do it using DNA, the same kind of process, although you use electricity to do it, where you put samples of DNA in a gel, apply electricity, and the DNA moves, but separates out according to the different size of those DNA molecules. And you end up with results that look like this. We'll talk more about that as we get to our DNA unit.